Welcome to the deep dive. So if you're working anywhere in small molecule drug discovery, you know the drill. It all starts with that one foundational problem.
Finding the hit, that very first compound.
Exactly. And the industry standard for that is high-throughput screening, HTS. But it's really starting to feel more like a bottleneck than a solution.
Oh, it's more than a bottleneck. It's a huge drain on resources. I mean, HTS is just brute force. You're testing hundreds of thousands, maybe millions of compounds, hoping something sticks.
And the success rates are, what, 1 or 2% typically?
If you're lucky. And that's for the easy targets. When you get to the really challenging stuff, the so-called undruggable targets, that number plummets.
We're talking about those really complex systems, aren't we? Like immune receptors or those tricky protein interactions.
Yeah, where the binding site isn't some neat little pocket, it's broad, it's shallow. For those, traditional HTS is often just a guaranteed failure, a huge waste of time and money.
Which is exactly why we're doing this deep dive. You brought in sources on 3 really advanced computational platforms that are designed to just shatter that old model.
Right. So our mission here is to get under the hood and understand how these AI and machine learning strategies are giving us a shortcut. And it's not just about speed. It's about efficiency, quality, and just the sheer scale of what we can now look at.
So the whole promise is moving from, just spraying and praying with chemicals to having the machine learning be truly well-informed. To have it tell us where the gold is buried before we even pick up a pipette.
Precisely. We need that precision, that predictive power. So we only spend those precious, high-cost lab resources on the candidates that have a real shot.
Okay, so let's get into the first one, HTS-Oracle. Because this one seems to tackle that undruggable problem head on.
It does, and what's so fascinating about it is how it understands chemistry. It's bilingual. It's a multimodal deep learning platform.
What does that mean in practice? Because traditional models have always had a hard time with the complexity of real-world molecules.
They did, yeah, because they often miss the context. So HTS-Oracle splits its analysis. First, it uses something called molecular language modeling.
Okay, so it treats the molecule structure, that SMILE string.
That long string of letters and symbols, yeah.
It treats that like a sentence in a language.
Exactly. It uses a model called ChemBerta, which is pre-trained on just tons of chemical data, to read that sentence and understand the context. It gets that a group over here can influence a group way over there. It's not just isolated words.
So it's seeing the whole paragraph, not just the punctuation marks.
You got it. That's the first stream, the context. The second stream is more traditional. It's looking at cheminformatics descriptors.
These like molecular weight, fingerprints.
Right. RT-Kit, fingerprints, log-P, all the reliable structural data. Then it fuses the outputs from both streams, and this is key, it runs them through an ensemble of 15 submodels.
15? That seems like a lot of computational overhead for one prediction. Is it worth it?
Oh, absolutely, because robustness is everything. You can't have one quirky model biasing your results when millions of dollars are on the line. And the proof is in the validation they did. They tested it on 3 really tough immune targets.
Right, TREM2 for Alzheimer's, CHI3L1 in cancer, and CD28 for autoimmune disorders.
Right.
This is where the rubber meets the road.
And the results were just stunning. For TREM2 and CHI3L1, HTS-Oracle cut down the number of compounds that needed lab testing by over 99%.
99%. Just think about the savings in time and materials alone. That's a total game changer for the project timeline.
It is. And it wasn't just about quantity. The quality of the hits was way up. They saw hit enrichment factors from about 8 times all the way up to an insane 176-fold for one of the screens. But the TREM2 case study really shows the quality improvement. HTS-Oracle didn't just find more hits, it found better hits. They identified a lead compound, 19BO3, that had a 3.5-fold better binding affinity than anything they knew of before.
So you're starting your next phase of development, the lead optimization, from a much, much stronger position.
A much higher baseline, exactly. And maybe the most impressive test was what it did with CD28.
Why was that one so challenging?
It was about the data. The training set was incredibly sparse. I mean, fewer than 2% of the compounds were actually active.
So the model had almost nothing to learn from.
Almost nothing, but it still managed an 8-fold increase in the hit rate. And it found totally novel compounds that could disrupt that critical CD28 interaction in autoimmune pathways.
Okay, so HTS-Oracle is about using deep context to find better hits for tough targets. It's about exploitation. Now let's pivot. What if your goal isn't just finding better hits, but finding completely new kinds of chemicals?
The novelty problem.
Yeah. How do you discover brand new scaffolds when you're starting with basically zero information? This is where ChemScreener comes in, the active learning approach.
Right, and active learning is a totally different philosophy. It's not a one-and-done search. It's an iterative loop, a conversation between the computer and the lab.
So the model makes some predictions.
On a very small set of data, yeah. Then those compounds get tested in the lab. The results, good or bad, are fed back to the model. It retrains, gets smarter, and makes better predictions for the next round.
It sounds like automating the scientific method, but it also brings up that classic tension, the exploration versus exploitation property.
Exactly. If the model only gets rewarded for being right, it's just going to keep suggesting things that look like the hits it's already found. You'll never find anything new.
So how does ChemScreener solve that? How does it balance playing it safe with taking risks?
That's the real innovation here. It's a strategy they call balanced ranking. And it's designed specifically to manage that tension. So it doesn't just predict how active a compound will be. That's the exploitation part.
The sure bets.
Right. At the same time, it calculates its own uncertainty for each compound.
Uncertainty. So the model is basically saying, I have no idea what this thing will do, but boy, it looks different from everything else I've seen.
That's it, exactly. And testing those high uncertainty compounds, that's your exploration strategy. That's where you find the novel chemotypes. The balanced ranking system just combines the ranks for both scores predicted activity and uncertainty to pick the next batch to test.
So it's forced to look at the most promising compounds and the weirdest, most interesting unknowns.
And the WDR5 case study showed it works. WDR5 is a target in aggressive leukemia. The original HTS screen had a dismal hit rate, less than 0.5%.
So basically a failure.
A total failure. But with ChemScreener, over 5 cycles, the average hit rate climbed to almost 6%, sometimes hitting 10% in a round. And more importantly, it found 3 completely new de novo scaffold series.
Proof positive. So active learning is the tool for when you need to intentionally push the boundaries of known chemistry.
Absolutely. So we have HTS-Oracle for quality, ChemScreener for novelty. And that brings us to the final and maybe biggest challenge scale, the sheer mind-boggling scale of the chemical universe.
Okay, let's try to wrap our heads around this. The 11 billion compound problem. Libraries like Enamine Real Space now have over 11 billion compounds that you can actually order and test.
And if you try to screen that with a traditional method like computational docking, it's just not possible. The math doesn't work. It scales linearly.
What does that mean in terms of time and money?
The source estimated it would take over 3,000 years on a single CPU, or cost you close to $1 million in cloud computing fees. A project would be dead on arrival.
So you have this massive, promising chemical space you can't even explore. That's where vSynthese comes in. Virtual synth hierarchical enumeration screening.
Right, and vSynthese is so clever because it breaks that linear scaling rule. It does it by exploiting the way these giant libraries are built. They aren't just 11 billion random molecules.
Or modular, right, built from smaller pieces.
Exactly. They're built from a much smaller, manageable set of core scaffolds and building blocks called synthons. vSynthese uses that modularity, that chemical logic, to avoid screening everything.
Okay, so walk us through the process. How does it sidestep the 11 billion compound trap?
It's a multi-step hierarchical process. It's brilliant. Step 1. They don't dock the full molecules. They dock a tiny fragment-like version. They call it the minimal enumeration library, or MELL. It's only got 600,000 compounds.
A tiny fraction, something you can actually compute.
Right. But step 2 is the secret sauce, productive selection. They don't just filter that MEL based on the docking score. They filter based on the pose, the way the fragment sits in the binding pocket.
Ah, so they're looking at where the next piece, the synthon, would attach.
Precisely. If that attachment point is pointing straight into a wall of the protein where there's no room for a bigger piece to fit, vSynthese just throws it out. It kills that entire family of billions of potential molecules right there.
So it uses the 3D structure of the target protein to prune the search space before it even generates the final molecules.
That's the leap in efficiency. Only the fragments that are positioned productively, pointing into open space, move on to the final step, which is where they actually build out and dock the full molecules. For that 11 billion library, they only ended up having to fully dock about 2 million compounds.
2 million versus 11 billion, that's, what, a 5,000-fold reduction?
Over 5,000-fold, yeah. It turns thousands of years of computation into a few weeks. It makes the impossible possible.
And the experimental results backed it up. The screen for cannabinoid receptors.
Yep, they got a 33% experimental hit rate. That's more than double what a standard virtual screen benchmark got. And the hits were good. They quickly optimized one into a highly selective antagonist with sub-nanomolar affinity.
Incredible. So this has really covered the whole spectrum. We've gone from, brute force searching to these incredibly intelligent targeted strategies.
We really have. And they each solve a different piece of the puzzle. HTS-Oracle is your quality maximizer. It uses that deep, contextual language model to find the best possible leads for the hardest targets.
And ChemScreener is the explorer, the novelty seeker, using that active learning feedback loop to push into unknown chemical space.
And finally, VSYanthese is the solution for scale. It tames those gigascale libraries by applying chemical logic directly to the screening problem.
But the common thread really is channeling those expensive, time-consuming wet lab resources. All three of these funnel your attention only to the most promising candidates, which just accelerates everything downstream.
It does, and that efficiency is only going to get more critical. And here's the final thought for you to chew on.
Okay.
The Anamine Real Library. It grew from 11 billion to over 21 billion compounds in just the time since this research was published. It's growing polynomially.
And it won't stop.
But because VSYanthese scales with the number of synthons, the building blocks, not the final library size, it's kind of made the idea of a too large library obsolete. So think about this. If we can pull subnanomolar hits out of 21 billion compounds today, what are we going to find when these synthesizable libraries hit the petascale? 1,000 trillion compounds. What discoveries are just sitting there waiting for us?